And welcome to Chapter 4. In this chapter, we're going to talk about congenital and hereditary diseases. The learning objectives of this chapter are several. First of all, you should be able to describe some of the more important malformations resulting from intrauterine injury. You should be able to list the common causes of congenital malformations and their approximate incidence. Describe some of the common genetic abnormalities and explain their methods of transmission. Describe the role of chromosome deletions and translocations in congenital disease. And list the causes of Down syndrome and describe its clinical manifestations. Give reasons why it is important to identify a carrier of a 1421 chromosome translocation. You also should be able to list 4 abnormalities of sex chromosomes and describe their clinical manifestations. You should be able to compare the methods of transmission and clinical manifestations of PKU or phenylketonuria and hemophilia. And explain multifactorial inheritance, and give an example of this and describe the relevant factors involved. You should be able to understand the various methods available to make a diagnosis of congenital abnormalities within a fetus. So, what is the difference between congenital and hereditary disease? Well, first of all, a congenital disease simply means that the abnormality is present at birth, even though it may not be detected until sometime after birth. With congenital disease, the original genetic code in the zygote is normal, but something occurs in utero that causes an abnormality within the genetic code. And again, this may be detected at birth, or it may not be detected until the person is much older. Versus hereditary or genetic disease, this actually results from an abnormality within the chromosomes or a defective gene that is present in the zygote. Causes of hereditary and congenital malformations, especially to congenital, is intrauterine injury to the embryo or fetus. Environmental factors. Chromosomal abnormalities in hereditary diseases. Abnormalities of individual genes. So, what are some of the causes of intrauterine injury? Well, certainly harmful drugs and chemicals that the mother may be exposed to. Radiation, both ionizing and non-ionizing. Maternal infections, such as measles infection, or rubella, or cytomegalovirus, or Toxoplasma gondii, which is a protozoan parasite. The 3rd to 8th week after conception, this is when the embryo is the most vulnerable to injury, as all of the major organ systems are forming during this embryonic period. We can actually classify drugs as harmful to the fetus into one of 5 categories. The first category is Category A, and these class drugs possess no risk to the fetus, and this has been demonstrated in well-controlled studies in humans. Category B drugs, there is no evidence of risk to the fetus. Either animal studies show risk, but human studies did not, or there are no adequate human studies, but animal studies do not indicate any risk. Class C drugs, a risk to the fetus cannot be ruled out. No human studies are available to assess the risk, and animal studies either are not available or indicate a potential risk. Class D drugs, this is where we now see positive evidence of risk to the fetus. However, the drug is needed to treat the patient, the mother, and no safer alternative is available. So with Class D drugs, we need to weigh the potential benefit to the risk to the fetus. And then Category X drugs, these are absolutely contraindicated during pregnancy. Severe risk to the fetus greatly outweighs any possible benefit to the mother. And there was a drug that was used, especially in Great Britain during the 70s, called thalidomide. And this was a great antiemetic drug. It actually helped tremendously with especially the first trimester morning sickness. The problem was that we begin to see a number of births with deformed limbs or missing limbs. And so we now understand that thalidomide is actually an X-class drug. Cytomegalovirus virus. So this is one of the herpes viruses. And we actually can see in this at the end of these arrows something called the misshapen cells here. And this actually are inclusions in the placenta villi. And the fetus actually died in utero of overwhelming cytomegalovirus infection involving both the lungs and the brain. Now, we can see abnormal number of chromosomes, and let me make this statement right now. It is better to have too much genetic material than not enough. However, it also depends upon how much you have and where the deletion or duplication is located. So oftentimes, abnormal numbers of chromosomes is due to a failure of homologous chromosomes in the germ cells during either meiosis 1 or meiosis 2 to separate correctly, and this is known as non-disjunction. Disjunction is the correct separation of these homologous chromosomes in meiosis. Monosomy is a situation where we have the absence of a chromosome within a cell. And so instead of having a pair of chromosomes at a particular pair, you only have a single copy. Trisomy, as the name says, means that we have 3 instead of a pair. So now we have the presence of an extra chromosome. Some of these can actually also be due to deletions where the chromosome breaks during meiosis and the broken piece is simply lost. Or it could be through translocation, where either a misplaced chromosome or a part of a misplaced chromosome attaches to another chromosome. And we're going to talk about translocation, especially in some of the cases of Down syndrome. So during the first meiotic division, the homologous chromosomes are all lined up along the equator of the cell during mitosis, where the spindle fibers are attached. And when meiosis 1 occurs, this pair will separate from this pair. And so we get this one down here and this one down here during normal disjunction. But what happens if both of these chromosome pairs actually goes to one of the daughter cells? That means we don't have any here then in the other daughter cell. And then we're going to undergo meiosis 2, where these homologous chromosomes now will separate at the centromere and should have one of each pair then going to this to go from diploid cells to haploid cells. But what if this occurred, where these two did not separate at the centromere and they both were pulled into one cell? Then this cell would wind up with 2 and this one would wind up with none. If these gametes were then fertilized by a normal cell, these cells would have one from this cell and one from the opposite gamete, having 2 in this pair, which is normal. But if this cell is fertilized or is joined with another normal gamete, I now have, instead of 2 pair in this spot, I now will have 3. And in this one, I only wind up with 1, and that one is from the other gamete. So this non-disjunction can either occur in the first meiotic division, where the homologous chromosome pairs don't separate from each other, or during meiosis 2, where the homologous chromatids do not separate at the centromere correctly. So let's look at the sex chromosome abnormalities. What if we had an extra Y with the normal XY pair? This is known as XYY, and really there's no significant effect because the Y chromosome mainly carries genes that's concerned with male sexual differentiation in the embryo. And so the Y only carries basically the SRY gene, and that gene determines maleness. So even though you have an extra Y, oftentimes we'll not see any significant phenotypic differences. However, because there is now 47 chromosomes, sometimes these individuals may actually have infertility because you cannot divide 47 equally. However, sometimes they actually will be able to produce normal sperm. What if you have an absent Y? Well, we do know that XX is normally a genetic female. But what if you just have one X? Well, the body conformation will be female because there is no Y carrying that SRY gene. However, typically the secondary sex characteristics in these individuals are highly underdeveloped. The other thing is now we only have 45 chromosomes. And again, we cannot divide that equally during meiosis. And so the vast majority of these individuals will actually be infertile. What if there's an extra X in the female? Well, there's actually, again, little effect seen in the phenotype because if we can inactivate one X as the lion body or bar body, then why can't we just inactivate the other X? Again, we now have 47 chromosomes, so this may actually cause some infertility issues. And what if there is an extra X in the male? So we have the situation of XXY. This actually has an adverse effect on male secondary development. Because of the presence of the two X's, most of the secondary sex characteristics will be feminized, even though this is a genetic male. And again, 47 chromosomes, so we can't divide them equally. So oftentimes these individuals will be infertile. So Turner syndrome. This is actually a situation where we have XO, and the O simply means there's no other chromosome in the pair. So we have a 45XO. And so this is actually the only monosomy that we see in humans that is survivable. Incidence occurs in about 1 out of every 2,500 genetic females. And again, fertility, they're oftentimes actually sterile or infertile, simply because 45 is not an even number. And again, I said it's better to have too much genetic material than not enough. Triple X syndrome is a 47 genotype with XXX in the 23rd pair. Phenotypically, don't see any changes. Incidence actually is probably pretty frequent, about 1 in about 850 live female births. Fertility is usually not impaired because, again, most of the eggs that would be produced, if they do have two X's in the 23rd pair, oftentimes they actually die before they could even be fertilized. And so only eggs with a single X in the 23rd pair can survive. Klinefelter syndrome. This is the XXY. So again, the presence of the Y determines that this is a genetic male. Incidence is about 1 out of 750 males. And usually, again, fertility is this individual is going to be infertile. XYY syndrome, genotype again of 47, but now we have XYY. Incidence in about 1 out of 850 live males. And fertility in this case is usually not impaired because normally we divide the X and the Y into the sperm. So even if a sperm is carrying two Ys, it really doesn't make a lot of difference. Now, another chromosomal abnormality that actually is found in the 23rd pair is something known as Fragile X syndrome, or this is also sometimes called X-linked mental deficiency. This is an abnormality of the X chromosome near its bottom tip, and it is actually a major cause of mental deficiency. There's an abnormally high number of CGG sequences in certain people's DNA chain at this point in the X chromosome. And because of the abnormally high CGG sequences, this causes a hypercoiling of the helix. This becomes more pronounced as it's passed down from parent to offspring. Women will pass this abnormality to both daughters and sons, but males will only pass it to their daughters in the fact that they can only give their daughters an X chromosome. Down syndrome. This is probably one of the most recognized chromosomal abnormalities in the world. It is caused by a trisomy of chromosome pair 21, and so this is an autosome trisomy. And this is either by non-disjunction, which is the most common form, or by translocation, and we'll talk about both. The most common chromosomal abnormalities in humans, as we see it in about 1 out of every 600 live births. Manifestations of Down syndrome, developmental disabilities, we can see cardiac malformations, major defects in other organs. Turner syndrome, went the wrong direction. Turner syndrome has no bar bodies because we only get the one X, it's going to be the one that has to function. In triple X syndrome, however, we just simply inactivate 2 of the X's. In a Klinefelter patient, one of the X's actually will be inactivated and form the bar body. And we get no bar bodies in the XYY syndrome because, again, we only have one X, and it's the X that has to function. So non-disjunction Down syndrome. Non-disjunction occurs during gametogenesis, and this can occur in either the egg or the sperm, but most often this non-disjunction actually occurs in the egg, and I'll explain why here in just a moment. 95% of all Down syndrome cases result from non-disjunction of chromosome 21 during oogenesis. Now, if you remember, the egg does not finish meiosis 1 until it is actually fertilized, and then it will finally complete meiosis 2. So these eggs are basically held in prophase 1, and as the mother gets older, her eggs get older, and what literally happens is that the chromosomes become sticky, if you will. And so oftentimes we get non-disjunction either as we complete meiosis 1 or as we go through meiosis 2. So the ovum winds up with the extra chromosome 21. 1 in 50 live births in offspring of older women older than the age of 40 are likely to have Down syndrome. Now, non-disjunction can occur when the zygote begins mitosis. And so normally, again, we're going to go from diploid cells to diploid cells, which that's fine. But again, sometimes we get a stickiness of the chromosomes, and we wind up actually then having 3 copies in one and 1 copy in another one. Better to have too much than too little, and in this case, since we're talking about an autosome, chromosome pair 21, the monosomy is actually lethal. Okay, this just shows then our normal mitosis that would occur. So sometimes this non-disjunction actually occurs after fertilization, but most often it is due to non-disjunction in the formation of the ovum. Now, translocation, about the other 5% of Down syndrome patients will actually be due to translocation. And this is where we do have an extra chromosome number 21, but it fuses with chromosome number 14 or another chromosome, and most often it is chromosome 14. So when we look at the number of total chromosomes, instead of having 47, we will still have 46 chromosomes because that extra 21 is actually stuck on 14. Causes of this, oftentimes there are normal chromosomes in the cell of both parents, and the translocation occurred accidentally during gametogenesis in one of the parents. And so we wind up with something known as a 1421 carrier in one parent. The carrier parent then only has 45 chromosomes because one of their normal 21s actually fused with 14. So if we have a 1421 carrier in one parent, the carrier parent is capable of transmitting that abnormal chromosome to his or her children, resulting in this translocation Down syndrome. Possible outcomes of pregnancy involving a female carrier would be the translocation chromosome is not always transmitted because she does have one normal 14 and one normal 21 chromosome. So that could be that the offspring is completely normal. Or it could be that the offspring becomes a carrier. Or it could be that the offspring is non-viable. Or the fourth outcome could be potentially translocation Down syndrome. And so this is what it looks like. So here we have a female carrier. So she's got a normal 14 in the black here, a normal 21, but her 14 and 21 of the pair are now fused together. So she has a 1421 chromosome. The male is normal. So the possible ova that this female could produce in chromosome 21, she could have a normal 14 and normal 21 plus the normal sperm. We're going to have normal 14 and 21 pairs in the offspring. If she actually puts the 14-21 combo chromosome in her egg and then mates with a normal male, the offspring will actually be a carrier. They'll have one normal 14, one normal 21, and the 14-21 chromosome. Or it could be potentially that neither the 14-21 or the 21 normal chromosome will be put into the ovum, and so now we don't have anything in the 21 place, and we wind up with a monosomy of 21, and this is non-viable in the autosomes. Or we could have the 14-21 with a normal 21, normal male, and so what we wind up with is a normal 14, normal 21, a normal 21, so we have a pair of 21s. We have a pair of 14s, but that third pair, or third of the triplet of 21, is now stuck on this 14-21 chromosome. And so if we look, we actually have 1, 2, 3, 4 chromosomes, but we have 1, 2, 3 copies of chromosome number 21. And this would be a translocation Down syndrome. Now, there are a few other trisomies of the autosomes. After trisomy of chromosome pair 21, however, oftentimes too much of any of the other autosomes can also be fatal in at least early life. Chromosome number 13, known as Patau syndrome, oftentimes in these offspring we'll see severe cleft lip, cleft palates, abnormal development of the skull and brain, abnormal eye development, sometimes actually seeing cyclopsia, congenital heart defects. And oftentimes, Patau syndrome offspring actually are polydactyly. Edwards syndrome, which is a trisomy of chromosome number 18, shows some of the same type symptoms, and both of these trisomies are usually fatal in the neonatal period or in early infancy. In fact, sometimes they actually result in stillborns or miscarriages. So let's look at some genetically transmitted diseases and how they're transmitted. So these diseases result from abnormalities of individual genes on the chromosomes. The chromosomes themselves appear normal. Some defects arise spontaneously, usually through spontaneous mutation, but others may be caused by environmental teratogens, which are agents or things that influence that cause physical defects in the developing embryo, such as, for example, the drug thalidomide. Mutations can occur then that are permanent changes in the genetic material. Again, they may occur spontaneously or in combination with exposure to something in the environment, such as radiation, chemicals, one of these teratogens, or viruses. Now, one of the ways that we see transmission of these genetic diseases is something known as autosomal dominant inheritance. So it's in it's in one of the first 22 pairs, and the allele that causes the disease is a dominant allele. Autosomal recessive inheritance, again, on one of the first 22 pairs, but now the allele is actually recessive. And so to actually see the disease in an individual, we would actually have to have both of the genes on the pairs inherited of the chromosomes to be of the recessive type. And a couple of these are phenylketonuria, and that should have an E in there, and Tay-Sachs disease. Codominant inheritance is when both alleles are expressed in the genotype, and each allele is expressed individually. A good example of this is someone that has type AB blood. So they have a gene for the A protein and a gene for the B protein on their pairs, and so what they're going to see is both the A expressed and the B expressed. Incomplete dominance is where both alleles are expressed, but now it is a mixture of the two. So no longer is it individual alleles. We see this actually in cattle. We can actually crossbreed a red cow with a white bull, and what we'll get is something called a roan. And so it's actually a mixing of the two colors that we see in incomplete dominance. And then we have the X-linked or sex-linked inheritances. And these are diseases that are carried on the X chromosome of the 23rd pair. Most hereditary diseases are transmitted on the autosomes, however. Very few of them, there are a few, such as color blindness and hemophilia, that are carried on the sex chromosomes. Now, multifactorial inheritance. This is actually the combined effect of multiple genes interacting with environmental agents. For example, the color of your eye is not coded for by just one gene for eye color. There are actually multiple genes that will code for eye color. And environmental agents will actually influence the expression of these genes. Congenital abnormalities such as cleft lip, cleft palate, cardiac malformations, clubfoot, dislocation, congenital dislocation of the hip, anencephalia, spina bifida, all of these are actually thought to be multifactorial inheritance, where something in the intrauterine environment interacts with multiple genes causing these defects. We can actually examine the fetal cells for some of these congenital abnormalities. And this can be a determination of biochemical abnormalities in the fetal cells, or we can actually simply look at the genetic makeup itself. So we're looking for chromosomal abnormalities. Does this fetal cell have 3 chromosome 21s instead of the normal 2? We can see biochemical abnormalities. Does this fetal cell have the gene that will produce the enzyme that is made to break down phenylketonuria, or does it not? We can analyze the DNA. And so how do we get these fetal cells? Well, oftentimes we're going to pick them up either from the amniotic fluid, and again we can also do biochemical testing on amniotic fluid, or we're going to use something called chorionic villi sampling. Ultrasound examination is detection of major structural abnormalities that can be either hereditary or congenital. So we can look for major abnormalities of the nervous system. Anencephalia literally means without brain. So the brain is not developed correctly, or at least the high brain is not. Spina bifida is actually an opening due to failure of the spinous process to form correctly in the dorsal side of the vertebrae, leaving an opening, and that can actually open to the outside. Hydrocephalus, having too much cerebral spinal fluid within the ventricles of the brain. Obstruction of the urinary tract, failure of the kidneys to develop, failure of normal limb development. And fetal DNA analysis, determination of the biochemical abnormalities by analysis of the DNA cells, will give us a lot of good information. And we can do this by amniocentesis again, or this chorionic villi sampling. Amniocentesis. If we take some of the fluid, there are several things that we do test for besides looking for just fetal cells upon which we can do DNA analysis. If we have high concentrations of the alpha-fetoprotein in the amniotic fluid, this is typically suggestive of a neural tube defect, such as anencephaly, microcephaly, or spina bifida. How do we take amniotic fluid? Well, we do it by doing a transabdominal amniocentesis under ultrasound guidance. Usually this is performed between the 14th and 18th week of pregnancy. The reason for this is the amniotic cavity with the fluid and the fetus is actually fairly large, and so we've got a lot of space between the actual developing fetus and the amniotic sac itself. The primary use for amniocentesis is the prenatal detection of chromosomal abnormalities in females older than age 35 due to, again, higher incident risk of Down syndrome in infants born to older females. Chorionic villi, this is the frond-like substance that is actually produced by the extraembryonic membrane that actually interdigitates into the endometrial wall of the uterus to form the placenta. So fetal cells or embryonic cells actually give rise to the chorionic villi. So the fetal cells are going to be obtained for evaluation using this chorionic villi sampling and not the amniotic fluid. A small catheter is actually inserted through the cervix to the site where the villus are attached to the uterine wall, and again this is done under ultrasound guidance. And then a small area is suctioned through that catheter to take for the sample. Now, advantages of chorionic villi sampling: it can be performed much earlier than amniocentesis, between the 8th to 10th week of gestation. It carries less risk it carries less risk from abortion if the parent decides to terminate the pregnancy in case of a congenital abnormality. So again, not as far along, going to be a less risk with abortion. Disadvantages: it's actually more difficult technically to perform than amniocentesis. Complications could actually be spontaneous abortion. We're going to disrupt a little piece of the placenta. Sometimes that little suction that we use may actually suck up part of the fetus at that point. So we could suck off an arm, which is not a good thing. So limb deformities, that sort of thing. So what are the consequences of chromosome non-disjunction? And can you tell me what the karyotype of an individual with Down syndrome is? Why did this happen? And what are the possible outcomes of a pregnancy involving a 1421 translocation chromosome carrier? What are the causes of congenital abnormalities and their approximate incidences? And what are the indications for amniocentesis? In other words, what is the primary reason that we do amniocentesis?